Okay everybody, in this video we are going to cover a brief introduction to transposable elements, also known as transposons. Now, okay, what are these things? Now, genetic elements, so a genetic element, that's, just a generic term for, a piece of DNA in the genome. Okay, so transposable elements are a type of genetic element that can move to new locations in a genome. So if they're found at one location, they can move to another one. So they do not have an independent form. Now, what do I mean by that? So they're kind of like viruses, how some viruses can insert into our genome, but the transposable elements can't exist outside of our cells or outside of our genomes. Viruses, however, they have an independent form. They can exist as viral particles outside of cells, and they can spread to new cells that way. So transposable elements do not have an independent form. So they can cause mutations. So that way they're kind of bad. And then that way they can be kind of bad. But at the same time, they can comprise much of a genome. And this is interesting, and we don't really know that much about why they can comprise so much of a genome. For example, in humans, 43% of our genome is made up of transposable elements. Here's another common example: corn, maize. 85% of the maize genome is made up of transposable elements. So much of a genome can be made up of transposable elements, and we don't know why. We don't know the reason for that. I mean, we do know that they can move to new locations and they can increase their copy numbers, but we don't know if these increased numbers form an important part of our genome. So in one way they cause mutations, but maybe all these copies form an important structural component of our genomes, and we don't quite understand that yet. So are there good things about transposons? Not sure about that yet. But we do know they can cause mutations and genetic disease, and we will cover one of those examples in the next video. So transposable elements can be divided into a couple different classes. In this introductory course, we can't cover all of the different classes of transposable elements, but we will take a look at one subclass of one class. So let's say class 1. Now these would be the retro transposons. These elements require a reverse transcriptase to move to a new location in a genome. And we've seen an example of a reverse transcriptase already in this class, right? Can you remember, you think about, in this course, I should say, where have we seen one of these? So telomerase, remember telomerase is a reverse transcriptase. In reverse transcription is using an RNA template to synthesize DNA. Right? Transcription is using DNA as a template to make RNA. Reverse transcription is using an RNA template to make DNA. And with telomerase, the RNA molecule, the RNA component of telomerase was the template to synthesize DNA to lengthen the telomeres. So retrotransposons make up class 1 transposons. Class 2, these are DNA transposons. And they use an enzyme called transposase. So they use an enzyme called transposase to move to new locations of a genome. So we are not going to talk about class 2 transposable elements. We're going to focus on class 1. And specifically, we're going to focus on a specific subclass of retrotransposons. So within class 1, there are 2 subclasses. The LTR retrotransposons, and they make up 8% of the human genome. And these are related to viruses like HIV. But unlike viruses like HIV, they don't have an independent form. So that's one subclass. Another, and LTR stands for long terminal repeat, if you're interested. Now the other subclass of class 1 transposons is the non-LTR retrotransposons. Now we're going to focus on this class, this subclass, the non-LTR retrotransposons. And why? Why are we going to focus on those? Well, because these make up 32% of the human genome. And if we only have time to cover, one subclass of a class of transposons, we may as well focus on the one that comprises most of our genome. And there are 2 types of non-LTR retrotransposons that make up most of our genome: line elements and sign elements. So line stands for long interspersed element. So there's the L, there's the I and the N, and there's the E for line. In sign, can you guess probably what that stands for? Short interspersed element. So lines and signs, and that's what we're going to focus on with our introduction to transposable elements. So there is one, and in this video we are going to talk about lines specifically. Then we're going to talk about a recent publication where they discovered a genetic disorder caused by a mutation caused by a line. And in the next video after that, we're going to talk about the signs. So now the most common line in humans is called L1 for line 1. So very simple name for line number 1. It's the most common line element in the human genome. 516,000 copies on average. There are 6 kb kilobase pairs long, so 6,000 base pairs long. Each is 6,000 base pairs long. Well, the full-length molecule is 6,000 base pairs long. As we will see, a lot of them are not full-length. And this element alone comprises 17% of the human genome. Okay, so what does it look like? So let's say this is a full-length L1 element in the human genome. It's about 6 kb long. And on either side, there will be these short repeated sequences. And the short repeated sequences have to do with the way it inserts into a new location. So these can be slightly different depending on where the element inserts into a genome. Now, this would be the +1 site for the full-length transcript. And there's another +1 site because there's an internal promoter somewhere in here that causes RNA polymerase to transcribe this way. And as far as I know, this transcript has nothing to do with transposition of the element. But this +1 site is important because this +1 site leads to the production of a bicistronic RNA. So an RNA that codes for 2 proteins. Let's say bicistronic RNA. And we covered this cistron term and when we were talking about, I believe, the lac operon and the trip operon. So essentially a cistron is just a part of a RNA or a genome that codes for a protein. So there are going to be 2 regions of the messenger RNA from the line 1 element that code for a protein. So this region right here, this would be the 5' UTR, 5' untranslated region. And this region right here, we are going to say is ORF1P. So this part of the line 1 element codes for a protein called ORF1P. I'll explain what ORF is in a moment. Then what we're going to have is a spacer region. And then we're going to have another region that codes for a protein called ORF2P. So this is the protein this region codes for. And we can just call this region ORF1 and ORF2. The proteins themselves are called ORF1P, ORF2P. Now this is the start codon for this protein, ORF1, and this is the stop codon, and here this is the start codon, and here this is the stop codon. So we have 2 proteins that can be translated from an RNA, from an L1 messenger RNA. And this region right here would be the 3' UTR, untranslated region. And then there's also usually a lot of A's in here. So you know how these messenger RNAs usually end with A's. Well, there are usually a bunch of A's actually inside the genome at the very end of the element. And you'll see why, when we go over the mechanism of how it inserts, you'll see why that is the case in a moment. So ORF, what does this stand for? So ORF stands for open reading frame. So essentially it's part of a genome where you can go from a start codon all the way down to a stop codon without there being another stop codon in between. So an open reading frame indicates a part of the genome that codes for a protein, and there are no stop codons in there. So if we were to start translating from, say, the start codon here, we would never hit a stop codon until we got to the end. So that whole region defines an open reading frame. So ORF1 is the first open reading frame. ORF2 is the second open reading frame. Now, this is the genome right here. So this is transcribed, and when it's transcribed, we are getting a messenger RNA, the poly A tail. We have the 5' UTR, the 3' UTR, and ORF1, ORF2 down here. So start, stop codon, start codon, and stop codon. So we can imagine ribosomes are going to load on here after this is exported, right, to the nucleus. And then ribosomes can load on here too. Now, I'm not completely clear if it's known whether this one ribosome will translate ORF1P, stop, and then stay on without disassembling and then start translating this, or if this is what's called an IRES, internal ribosome entry site. I'm not really sure. You don't need to be sure either. I think that's something maybe for future iterations of the course. We'll take a look at that. But what we need to know is that ORF1P, once this is translated, we're going to get a protein called, well, ORF1. Once it's translated, we're going to get a protein called ORF1P. And what ORF1P does, it's an RNA binding protein. It binds RNA, and specifically it has an affinity for the line 1 messenger RNA. So it binds the line 1 messenger RNA. ORF2, this is a bigger protein, and it has a number of activities. Most importantly for our discussion today, this is ORF2 protein. This is the ORF2 protein. It has a reverse transcriptase activity and an endonuclease activity. So at least 2 activities there. And there might be some others. The endonuclease activity, now this is going to be important for making a nick in the genome at the target site. The target site being the site it's going to integrate into. Okay, so we have our 2 proteins that are key to the transposition mechanism. And as we go over that, make sure we're still recording. We are. Okay, perhaps it's best to, envision a chromosome right here. And let's imagine that there's an L1 element right here. And this L1 element is transcribed into a full-length L1 messenger RNA. So briefly, 7-methylguanosine cap, right? This is eukaryotes, so this messenger RNA is going to be capped. It's going to end with a poly A tail. ORF1 region, ORF2 region, 5' UTRs, 3' UTR. So this is going to be exported to where? Exported to the cytoplasm, right? Because that is where translation takes place, right? So the L1 element inside the nucleus on this chromosome, let's call this chromosome 1. Maybe it's on chromosome 1. It's transcribed by RNA polymerase II. We have the L1 messenger RNA, which is processed, cap, poly A tail. There are no introns that I'm aware of. Exported to the cytoplasm. So in the cytoplasm, ORF1 and ORF2P will be produced. And what will happen is they will bind the messenger RNA. So after this messenger RNA is translated and the proteins are synthesized, what happens is, let's say these little guys here, these little things, these are representing ORF1P. So many copies of ORF1P binding to the messenger RNA, the L1 messenger RNA. And say down here, the poly A region and the 3' UTR, well, that's where ORF2P is binding. Now, when they bind to that messenger RNA, they have the ability to, let's say, import maybe a better word is bring this L1 messenger RNA to the nucleus. So the messenger RNA was exported from the nucleus to the cytoplasm, so these proteins could be made. Once the proteins are made, they bind to the L1 messenger RNA and they bring that messenger RNA back into the nucleus. Okay, so then that messenger RNA is used as a template to make a complementary DNA molecule. Now, in order for that to happen, a target site must be chosen. And the target site choice is, I'm going to say, chosen semi-randomly. Target site. Can you see that? Yes. Okay. Semi-random. Target site chosen semi-randomly. And I'm saying semi-randomly because there is a loose consensus sequence of T, 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 A associated with L1 insertion events. So what does that mean? Okay, so if this is the 5' end of a DNA molecule somewhere in the genome, this is a 3' end, 3', 5', and I'm going to put my T, 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 A right here. That site right here between this T and this A, this is where the nick occurs. So ORF2P has an endonuclease activity, so it breaks the phosphodiester bond between those two bases right there, typically. Okay, so what else is here? Well, it doesn't matter. The other bases don't matter so much. I'm going to put a T here, A. Those others don't matter so much. Okay, so this is where the nick is going to be made by the ORF2P that's holding on to the L1 messenger RNA. And that's going to allow for a process called TPRT for target primed reverse transcription. Oh, so pretty complicated term there. But why do we need this? Why do we need this? So reverse transcription requires a 3' OH group in order to, start the transcription process. And it turns out, I'm going to show you how the target actually serves as the 3', provides the 3' hydroxyl that primes reverse transcription. So assume that nick has been put into the backbone there, into the DNA, that target location. And here's the DNA molecule. So that nick is made here, and then somehow this is unwound a little bit. And keep in mind, this is a model, so I don't know how much is really been exhaustively tested and shown that it must work like this, or we just have a slight bit of evidence that this is what happens. But this is the current model we're going over. And there are going to be some A's up here, right? Okay, so this was nicked and slightly unwound. Now that poly A tail of the L1 messenger RNA can bind to these T's by complementary base pairing, right? And so we have a 3' OH, that 3' carbon of that last thymine nucleotide here has an OH group on it. And here's the L1 messenger RNA now, and now it's annealing. The poly A tail is annealed to those T's. And then I'm just going to diagram the L1 RNA like that. So this is the L1 messenger RNA. And remember, it's got, probably has the ORF1P bound all along it. And now this is where that reverse transcriptase was, down here. It's probably holding on to that poly A tail. It nicked this guy right here. And I'm diagramming it like this to show you that, okay, now it can start synthesizing. It can use this T to copy the RNA molecule, using the RNA molecule as template to make a DNA. And this is the 5' end of the RNA. This is the 3' end. So the transcription's happening from the 5' end here in the 3' direction. So this DNA, this is DNA. This is the RNA template. This is the DNA. We're going to call this the cDNA for complementary DNA. And I could show these hydrogen bonds formed between them. So this is this molecule right here is an RNA DNA hybrid. Okay, so once this is made, this is kind of a weird looking structure, right? Now, if you're a little confused, I don't blame you, because it's so confusing. How can this happen? How can ORF2p hold on to the DNA, make the nick, and then move along this RNA template? And then now, how is this piece going to get put into here? And what about this RNA? What's going to happen to that? So a lot of confusing stuff going on here. So essentially, this was the target site. And here's the L1 messenger RNA, cDNA copy. So an enzyme called RNase H is used to chew away that L1 messenger RNA and to leave us with something like this. So here's the L1 cDNA. And now we have some unknowns, and it's my understanding that these aspects of the mechanism are very poorly understood. Essentially, what we need to do is link this up here somehow. And so let's say that's part A, linking this up to this up there. Part B is nicking this strand up top to make room for this sequence right here. So nicking up here, and the nick is thought to occur right here. And the reason the nick is thought to occur here is because if you remember in the very beginning of the video, I said there were some duplicated sequences. The genome was duplicated, tends to be duplicated. Very short duplications exist on either side of the element. And it's thought to do with the fact that this nick of the second strand of the target site probably occurs somewhere 3' of the first nick. So if the first nick was here, the second one's probably downstream a little bit. And then C, gaps need to be filled, gap filling. Right? Because if you put this in here, there's no place for it. You're going to have to open this up, and then there's going to be some gaps. And those need to be filled by DNA polymerase. So after that happens, the important part, if you understand the first part, the target primed reverse transcription, and up to the RNA-DNA hybrid connected to the target site for the L1 transposition mechanism. If you understand all that, that's good enough. You don't have to worry about those unknown steps, because no one really understands those. I'm sure there are theories or hypotheses on what happens, but like I said, I don't think there are any good explanations for how that process works downstream of reverse transcription and formation of that RNA-DNA hybrid. But okay, let's go back to the very first diagram for the mechanism. I said, okay, imagine the L1 is right here on chromosome 1 at this location. Now, this is the original location, ignoring the fact that there are like 516,000 other copies. So we're just curious about this one. This one was transcribed. And now, after the mechanism, it exists at a completely different location, a new location. Let's say this is where it integrated. Let's say this is chromosome 2. So now we have a new L1 element at a new location. And with these retro transposons, they're still at the same location. The original one is still there, and now there's another one. So let's say this is a genome that's never seen L1 before. and it just, for some reason, there's one site there. Well, now it existed in one copy before, now it exists in 2 copies. So you can see that's probably how these numbers go up inside a genome, because they just keep making more and more copies of themselves. So it increases by, well, the number increases by 1 every time it moves to a new location. Another important thing is new L1s often lack 5' end. And an explanation for that is if reverse transcription terminates, right, before reaching the end of the L1 mRNA template, right? If it terminates early, which might happen, then the whole L1 won't be copied into the new location, and it will be truncated, and that truncated L1 won't be functional. So in the next video, we're going to take a look at a relatively